Okay. So we go back and so we stick to the last version of the agenda, and so we start with a common session with Daniel and Laurent, and we start with Laurent about phosphate.
Thanks a lot. I will try to hurry up a little bit because I was not sure about the timing. There was some possibility of change. So I'm going to speak about phosphate. Just for starting. Okay. So just to remind you that phosphorus is one of the essential macronutrients. So you can see the difference of growth in plant on crops, which receive fertilizer and without fertilizer. And the second important point is phosphorus is a finite resources. So we extract phosphorus from rocks and we expect to reach half of the consumption in the next 2 or 3 decades. Half of the soil around Earth are lacking phosphorus to get proper development of the plants. Another little point is phosphorus came from rocks, mostly from sedimentary origin. And this is one of the problems because in the sediment offers a co-precipitate with metals, which can be toxic and which are provided in the field. Driving regulation. Why it's important for plants? Because you find this component in energy-rich molecules like ATP. It's crucial for photosynthesis and respiration. It's a constituent of genetic materials like DNA, RNA. It's also found in component of lipid bilayer with phospholipids. And it's also a very important component for signal transduction like phosphorylation. And when we speak about phosphorus, in reality, the plant can absorb phosphorus only on one way as phosphate, which is the main source and the essential source of phosphorus for plants. So I just provide you, we recently developed in collaboration with Chinese collaborators, a way to stain in tissue phosphate and only phosphate, not phosphorus. So you may be interested, this is rice, and you can see this is a semi-quantitative techniques. It's give you deposition of phosphate at cellular level. It's quite precise. It's semi-quantitative. You need to have measurement because it depends of the time of staining. So if you are changing the time of staining, it will be increasing the intensity of the signal. So it's quite good to visualize phosphate distribution in plant. The problem, it's not very discriminant because we are in liquid. So soil composition are mostly here. So you can see for plant growing on soil, it's not very discriminant. And it's only semi-quantitative. It was just published 1 month ago. I just want now illustrate the phosphate mobility in soils. So you have here 2 crops growing in soil or in water. And we use a technology developed in Japan to image radioactive phosphate using a scintillator coat on the objectives. So when the radioactivity provided by P33 or P32 hit the scintillators, it's transformed into photons and light that can be recovered by high-sensitive camera. So if you provide the same amount of radioactivity and you mix with the soil or the water, you can, with the camera, record over time the entries in the leaves of the plants, and you can see that the soil-growing plants absorb very inefficiently phosphate compared to plant growing in liquids. And actually, it's important because if you grow plants in liquid compared to plant growing in soil with the same amount of phosphorus, in liquids, it's 10 to 50 times more efficient to recover it. So to get the same phenotype, you need to increase the amount of phosphate in the soil by 10 or 50 times. Interestingly, if you provide phosphate, we provide this on the tip of agarose and on the root of the plant, you can see that it enters the plant and will be in all over the plant within 20 minutes. So when the phosphate reaches the plant, it's mobilized and distributed very fast. So I just want to remember that there is a very poor mobility of phosphate in soil, mostly because it will precipitate with organic matter or cations. If you have the same phosphate concentration between liquid and soil, it's a lot more efficient in liquid, and you have a very fast mobility in plants thanks to high affinity phosphate transporter from the PHT1 family. So we characterize, and we were the first team to co-publish with American the first mutant for phosphate transporter in plants. And interestingly, it's regulated at a lot of different levels, at the level of transcription, at the level of the transport to reach the membrane, at the level of the membrane with recycling or degradation. So we make many publications to describe this. And I just want to illustrate one of these mutants that we identified a long time ago. You have the phosphate transporter fused to GFP. So you see a cell with a phosphate transporter on the membranes. And in the mutant, which affect the targeting, the protein is very inefficiently transported to the membrane. And compared to a wild type, the mutant is lacking more than 50% of the delivery to the membranes. And we use these tricks for a little project I will present you. Because when you are introducing phosphate, this is a radioactive solution provided to plants, you see that the root tips are highly labeled. It's quite obvious. And if you provide phosphate only at the middle of the root, very quickly, within 30 minutes, again, you find the root tips labeled. And the question is simple. Why is it labeled? Is it due to uptake at this level or translocation? Because I remind you that the growth of the root takes place at the root tips where there is a meristem. So it's normal, you need phosphorus to make DNA and cell replication. So just to solve the problem, we use the same imaging system coupled on a microscope to look at the root tips. So you can see, you can follow the absorption of phosphorus on a wild type and on the mutant. And interestingly, on this time course, mutant responds very differently from wild type. If you are waiting longer, it's growing. So you will label the root tips, but not in 10 minutes. And we complement the mutant only in the root cap. And you can see that we can restore completely the import of phosphorus, demonstrating that these teeny layers here, which is a few microns, is able to uptake 30%, up to 30% of the phosphorus. Please don't keep in mind that phosphate enters plants by root tips. A fraction enters by the root tips, but this fraction is sufficient to trigger all the regulation of adaptation of the plants. So what's happened when phosphate arrive? We have two kinds of answers on plants. The first one are local. It means that if you put phosphate here and not here, you will have a difference of response of the roots which have received the phosphate compared to the one which have not received the phosphate. Actually, if you transfer a root from a rich media to a poor media, within 30 minutes, you can start to see a difference of growth. And compare, this is affecting the root, primary root growth, the meristem, lateral roots development, and root hair formation. So you can see the aspect of a root growing in rich media compared to poor media, which are pretty different. The second kind of response are called systemic because if you provide the phosphate, it will move very efficiently into the plants and trigger the answer all over the plants. And this will modify a lot of response, mostly at the transcriptional level. And we are identifying them and conduce the first identification of the gene regulator affecting phosphate recycling. For example, you are going to change phospholipids into galacto or sulfolipids, so trigger novel way of response. Phosphate recovery, you are going to secrete phosphatase to recover phosphate from organic matters, or phosphate uptake and transport. So just one example, we identified in the gene responding to phosphate presence. So those genes are highly induced by phosphate starvation. And when you add phosphate, you can see they are repressed within a few minutes. Actually, we start to have a really significant response after 30 minutes. So we identified many markers responding in root and shoots, and we use them as molecular marker for a project. I'm sorry, Philippe Chaume has left because it was an impulsion project, and with Philippe Sies. So we try to identify molecular marker in plants, and we develop this in different crops and model plant Arabidopsis. So I present you wheat grown with different level of phosphorus. Sorry, it's you will have the different amount of phosphate. I don't know why the different amount of phosphate used. And you can see that the fresh weight increase only during a limited amount of supply after you reach a maximum. And when you reach a maximum of development, interestingly, if you look at the phosphate accumulation, you accumulate phosphate. But here, where phosphate is really limited on the development, of course, phosphate is used, so it's not stored, and no way to measure the phosphate to have an idea of the state of starvation. Just for yourself, maybe you know the 75% of the phosphate absorbed is done during the first quarter of growth of the plant, really at the beginning. So we try to develop markers based on the one we identified. And so the response to starvation, when we provide phosphates, they are strongly repressed and they are discriminant in this amount of phosphate. So we can use them to say those plants need to receive phosphate or not, and in the amount where it's important, where the growth is requested. So we just validate those molecular markers and publish them last year. And we have validated also that you can use them on plant growing in field and detect if they are lacking or not phosphate. Because a little tricks, if you measure phosphate in soil, you can measure total phosphorus, but it's not the phosphate which will be absorbed by the plants. The available phosphate is very difficult to measure and need radio tracers. The problem of the molecular markers with RTQ-PCR is you access to the total pool of RNA. And the total pool is equilibrium between synthesis and degradation. It means that if you provide phosphate and the signal transduction start, you don't see them because the half-time of the RNA is more than 1.5 hours. So the transcriptional control can occur, and you will see delay if you are looking at the total pool of RNA. So we developed imaging of transcription in collaboration with a CNRS colleague, Dr. Edouard Bertrand in Montpellier, by fusing a protein to GFP, and this protein can recognize specific RNA loops. So if you introduce these RNA loops in the molecular marker I described you, they will agglomerate those proteins, and you will have a big spot of fluorescence because for one RNA molecules, we can agglomerate until 500 molecules of GFP. And what you see is this. Now, the image is pretty poor quality. You have transcription side and individual RNA. I'm sorry, the image magnified low quality. It's better here. So this is transcription site because we have many ribosomes synthesizing multiple RNA molecules at the same time, and you don't see it because resolution is too much light. You can see even individual RNA molecules. So if we couple that with microfluidics and spinning disk microscope, which can take a few hundreds of images, we can image transcription in real time in the cells. And we use that to demonstrate that the signal which was received by the plants affect transcription within 3 to 5 minutes. It means that when you change phosphate concentration, the plant responds within minutes. So I will finish here and thank all the persons involved in the works that have been presented. And I want also to say, if you're interested, we are working now on the signal transduction and coupling of phosphate and nitrogens.
We will switch to Daniel, and you may ask a question afterwards.
Hello, can you hear me?
Yeah, talk a little bit more loud.
Is it better now?
You can share your screen, Daniel, if you want.
I uploaded, I can, I
Okay.
find it, I find it. I have it, I have it. Is it good?
Yes.
Yep.
So hello everyone. Unfortunately, I have a flu, so I couldn't come to the lab today. But I'm glad to present some work I did together with colleagues here at the lab, Philippe CA, Kato Tanaka, and Jan Goucher, on work on enhanced weathering, in particular on its side effect related to phosphorus, which is the good stuff for plant, as we just learned from Laurent. So why do we need to remove actively CO2 out of the atmosphere? That's because you can see here in the black line the net CO2 emissions, which would be consistent with a 1.5 warming degree, a global warming of 1.5 degree by the end of the 21st century. And you can see that we need carbon dioxide removal from the atmosphere, in particular as current policies are not in line with the trajectory needed to reach this warming. You can see here the star, which is the greenhouse gas net emissions according to current policies for the year 2050, which is roughly 30 gigatons of carbon, but we should rather be closer to zero net. So we need some solutions to remove CO2 from the atmosphere because societies fail to cut their emissions. The different technologies proposed to do so, BECCS and afforestation are the two main measures which have been investigated quite a bit in models. However, they have limited potential for CO2 removal with respect to afforestation, or with BECCS, they are just not yet in place. So there are other solutions being investigated. And then today I want to talk about enhanced weathering, which has a couple of aspects which makes it quite attractive. Enhanced weathering is the use of rock dust. It's spread on the surface. It removes CO2 abiotically. It has positive side effects on plants and ecosystems, or positive side effects on the carbon cycle. It can be co-deployed, so you don't need to have, you can cut down a forest to have your CDR. You can apply it to agriculture or forest land. And the rock is cheap and it's abundant resource. And this reaction by which CO2 is removed by rock dust is basically an acceleration of a natural reaction of silicate minerals with CO2 in the atmosphere. This uptake of CO2 on a natural way is very, very minor. It's also called the Earth's thermostat because it balances the CO2 concentration on timescales of millions of years. Basically, it removes the CO2, which is outgassed by CO2, and balances it. However, if you take the rocks, you increase the specific reactive surface and you spread it over a large area of land, you can theoretically accelerate that process by several orders of magnitude and thereby introduce a carbon uptake which is substantial enough to counteract current disturbance of the carbon cycle by human activities. On top of that, it has side effects. One of them is it improves soil fertility. On the one hand, if you add rocks to the soils, some rocks like basalt, they contain potassium or phosphorus. And on the other hand, also they have some kind of liming effect by which they can enhance the availability of nutrients already present in the soil, but not directly bioavailable. So it overall improves also soil fertility. So it's an incentive for farmers to apply this technology on their fields so they can reduce their fertilizers and also cash in some money if they get money for reducing carbon dioxide from the atmosphere. And on top of that, in agriculture, you have already the infrastructure there to transport and apply rock dust. So you have the streets going to the fields, you have the tractors, and you have machines which can spread dust. So the infrastructure is there. And a study of a group of David Bierling in the UK, they showed here on this plot that the lines here is the phosphorus which could be released by basalt application in the UK, and the range with these red bars, they give you the crop requirement of phosphorus. You can see that for high application rates of basalt within the next decade, you could actually provide enough phosphorus for the crops to maintain yields by cutting down on the mineral phosphorus fertilizer. But what about application in forest ecosystems? You know, there's only a given area of cropland. Can we also apply rock dust into forest ecosystems? Here you see results from studies from colleagues in Hamburg, where we looked at how much phosphorus is being exported in forest in the US by timber, by harvest of wood. And in the dark lines, you see how much potassium or phosphorus could be supplied by rock dust. And also here in forest ecosystem, it would be possible to replenish the phosphorus being taken away by the fiber harvest when using basalt. So the other question is, what if we apply it in a natural forest ecosystem, or degraded forest ecosystem? However, because it's technology with unknown side effect, we first tested it out in a model before it's actually done in a field because of the unknown side effects. So what we did is we used Orchidee model that Philippe Pelor already presented to you earlier. We used a version which included the nitrogen and phosphorus cycle. So tree growth is constrained by water, temperature, CO2, but also nitrogen and phosphorus. And we did idealized experiments where we spread all remote forest areas by basalt using an airplane. And we looked how much CO2 is being drawn down by the chemical reaction, by the abiotic enhanced weathering reaction, and how much can be taken up by stimulating forests by giving them some extra phosphorus. So we did global simulations, and here you can find the map of the accumulated CO2 being removed over a time scale of 50 or 60 years, I think, for an application of 3 kilograms of basalt per square meter, something like that. And you can see there's very heterogeneous uptake rates globally, but we see some hotspots in the Amazon basin and also in Siberia, where we have quite large CDR removals, quite higher CO2 removals. And we also looked to what extent this removal was contributed by the reaction of forests to the addition of phosphorus. And this is shown here. The more greenish the plot, the more of the CO2 being removed was by enhanced tree growth versus the abiotic reaction. And here we showed for the first time that this side effect of basalt can actually be as large or larger than the enhanced weathering reaction. We also looked if this is enough to reduce the pressure on land needed for BECCS, for bioenergy plantations. So we used here not Orchidea, but a simple coupled climate, human energy, and land surface model. So it's basically an emulator of more complex models. And we did simulations for a 1.5 warming scenario, where we assume that by the end of the 21st century, a warming of 1.5 degree is reached. You can see this on the top plot. And we had different scenarios, one where there's only BECCS. So that's usually the usual scenario applied in IEMs. For example, in this IPCC reported 1.5 warming scenario, I think BECCS and afforestation were the only two measures to remove CO2 from the atmosphere. So that's the conservative standard scenarios in most models assessments at the moment. And then we did simulations where we include enhanced weathering in brown and enhanced weathering accounting for this phosphorus effect, which is a bit uncertain, which is quite uncertain at a global scale. And what we found is that this will not help, the enhanced weathering alone is not sufficient to keep warming below 1.5 degree, but it can help to reduce the amount of BECCS we need. It also allows for a larger overshoot. You can see the temperatures here, they're overshooting the warming target because it's close to unrealistic that we actually stay during the whole period below 1.5 degree warming. And also in the lower plot, you can see it can help to reduce the costs of this carbon dioxide removal. So it seems to be a valuable measure within the portfolio of CO2 removal technologies, but it cannot be the single one solution. And the current state of enhanced weathering is that it has not reached a commercial technological readiness level. Mostly modeling studies suggest that there's a substantial CDR potential. For example, France could offset all greenhouse gas emissions from croplands if they would apply a substantial amount of basalt on all French croplands. So it can really make a difference. At the moment, several field trials have been initiated because we don't have yet field proof that this actually works well. We only have proof for mesocosm and idealized dissolution experiments. These experiments in the field, they're restricted to a few years, have mixed results, so it's not quite clear. But on the other hand, there are several startups which already put tons of basalt out at the moment, which really take over these field experiments by trying to commercialize the whole application. However, the downside at the moment is that there's largely unknown side effects. There's a few studies just emerging or in preparation which show that the response of soil organic carbon is quite large. And as you know, soil organic carbon is the largest carbon stock on land. So if it responds in a way that it increases, good for us. But if SOC is being released from soil, this would be really bad because once you apply basalt to the soil, it's impossible to remove it again from the soil. There's also a problem of heavy metals contained in the basalt, among other different side effects with most often with biota, with soil biota or plants. And overall, the monitoring, reporting, and verification of enhanced weathering is quite challenging because it involves carbon changes in organic and inorganic forms in the soils, in plants, in rivers, and in the ocean. So I guess it's one of the most complex MRV challenge among all the different CDR technologies. Yeah, if you're interested in that or you want to learn more, write me an email. I also have here a QR code of my webpage where we repeatedly put news about new papers from the team. With that, I thank you for the attention.
Thank you.